Because opposite charges attract and like charges repel, pH-dependent changes in surface charges influence the conformation of biochemicals such as DNA and proteins. Conformation of these molecules has a large effect on their functioning. Organisms have sophisticated mechanisms to maintain cellular pH within narrow limits and prevent conformation changes that might interfere with the proper functioning of their biochemicals. The pH also influences biochemical energy storage. Ions pass through biological membranes only slowly if unaided by protein pumps, channels, or carriers that traverse the membranes. Cells of most organisms exploit this low membrane permeability to store energy. When surplus energy is available, they pump protons across the membrane and establish a proton concentration gradient. To retrieve this energy, cells let these protons flow back across the membrane through integral membrane proteins called proton ATPases. The flow of protons through a proton ATPase produces the high-energy compound ATP. The interplay between structure and function of proton ATPases is fascinating. As protons flow from a compartment with a high concentration of protons to one with low concentration, they pass through a channel in a protein that causes several subunits to rotate. Some of these subunits form a complex with ADP and phosphate, and as they rotate, conformational changes in the subunits squeeze the ADP and phosphate together and generate ATP. This process is analogous to placing a water wheel in a flowing stream and using the resultant rotational motion of the wheel to run a press that makes high-energy bars.